Okay, so morning everyone from Chicago. I think it must be pretty, I mean, it's not morning at your place. But anyways, uh, for those of you who don't know me, I was a student at ISERC. I think I passed 2 years back. And presently, I'm doing my PhD at University of Illinois at Chicago, and I work on disorder treatment. I'll come to what I do at the very end, but I'll talk mostly about like what is this. I think I have tried to make it as simple as possible, but in case at any point there is something that you understand, please talk. And that's the whole objective, like to get as much as possible. I have tried to include only the basic fundamental principles of disordered protein. So yeah, let's see how it goes. So I believe most of you are already familiar with the structure of protein and what they do. So in principle, like in very fundamental, if you think like that, so what are proteins? They are basically essential nutrients in our diet, right? That's what we know when we are very small. We know that we need to eat certain kind of food that has protein so that it's good for our health. Uh, Now, if you look more closely, you will see that protein is made up of a sequence of amino acids, which is something like this. Use a pointer. Uh, So, if you check, this is the primary sequence of a protein. It just contains a series of amino acids. Next, if you like, what happens is some part of this sequence might give rise to some second like alpha helix, beta sheets, turns, loops, etc. So secondary structure is basically when a small part of the primary sequence folds to give rise to some structure. So this is a typical example. Secondary structural elements can fold to give rise to a folded 3D structure, which may look something like this. So you see, you have the helices, you have those beta sheets, and they kind of fold into a compact structure to give rise to the tertiary structure of a protein. And then we like this is what we learn in our very first understand like very first courses about proteins, that the structure gives rise to function, something similar to the lock and key hypothesis of enzymes that you all have. I think if you have studied biology, you must have read it in class 12, that in order for an enzyme to fit into an active site of a protein, it must have a predefined shape. So what it means that if you want if you want a protein to function, you need to have a particular shape. So in that case, so let's say you have a protein in a predefined shape and you want it to bind it to something. So if that shape is not there, the binding will not occur. So this is the primary sequence, like this is the basic structural overflow of how a protein in general is seen, from amino acid sequence to a structured element. I see a hand.
Yeah, it's me. Uh, Hello.
Yeah, I can hear you. Tell me.
Yeah. So, what is the difference between this alpha helix and beta sheets? I see that they both are in similar shape, but only we are viewing from some different angle.
Oh, so this it's just the way how they look. So they an alpha helix is formed. You see, it looks something like this. This is a helix. A helix is
Yeah.
formed when like protein of, let's say, the IF protein, it forms a bond with the I plus fourth amino acid. So that's why it
Mm-hmm.
gives rise to this curly kind of representation. Whereas beta
Mm-hmm.
sheet is kind of a parallel arrangement of amino acids, which in which they have in like, so you see, this is a series of amino acids, this is a series of amino acids, and this is also a series of amino acid, and these are stabilized by hydrogen bonds have in like in these areas. So that's what
Okay.
I think. There is a small beta sheet here, you see, here and here. So if you have a beta sheet, that essentially means there will be another in parallel. You cannot have one isolated beta sheet. There will, so you see, if this is a lysozyme, and you have one beta sheet here and one beta sheet here, so that they can form these parallel hydrogen bonds. So that's another kind, that's a kind of secondary structure.
Okay. Is there any significance of this helix kind of thing in those seeds? Like some helix kind of thing we can see.
Here,
Some helix kind of thing is there. Some curves are there. So is there any significance of those
which one do you mean, like
in the betas in the figure schematic of that beta seeds? There are some.
let me check. Yeah, tell me where. So this is the beta sheet. Now, which one are you saying?
Mhm.
Is helix in behind?
Uh, uh, in the second figure of the first row, there is a beta sheet. You have drawn some schematic of that. So
This
there are
one?
some helix kind, yeah. In that some helix kind of thing are there. Is there any significance of those or they are
Oh,
just
this in the in like this one?
yeah, yeah, this one.
No. That's just a representation of how one beta sheet is connected to another. I mean, this is just a cartoon representation. So you see, this one
Mm-hmm.
goes from here to here, then from here to here, and like that. It's just a
Okay,
cartoon representation, nothing else.
okay. Yeah, thank you.
Anything else? Then I think I can move on next. So from here, what's needed as we move forward is the basically the primary sequence, that's the amino acid sequence of the protein, and how it can fold to give rise to the to the secondary structures. So next, let's see that how many types of amino acids are there. So in general, like naturally occurring amino acids are 20. You can of course generate more. There are synthetic amino acids as well, but that's not the point. Uh, yes, so
Souvik Dey, I was asking that you told there are two types of representation are there, alpha helix and beta sheet.
there are
So
more
is,
turns, loops. Yeah.
yeah, turns, loops, and all other things are there. So how do we determine that how the protein will form the structure? Which will go for alpha helix, which will go for beta helix?
So one thing you can look at the sequence. So there are certain proteins, I mean, sorry, certain amino acids that favors alpha helix, and there are some which like breaks alpha helix. For example, glycine and proline, they are pretty well known to like,
Mhm.
for example, proline are pretty well known to break an alpha helix because they have so if you look at the structure of, if you look at the structure of proline, they have a very rigid say rigid side chain. But on the other hand, if you look at glycine is very simple. It has only one hydrogen in the side chain, so it's very easy to fit in. So basically, you have to look at the amino acid sequence, and there are predictors that have been developed by looking at the sequence of amino acids, and that can help you predict that if it will form an helix or if it will form a beta sheet.
Okay,
For example,
thank you.
like if we start from a random protein structure and we work with disordered protein. So I'm coming towards that. But what we start with is so there are online servers that will help you predict the secondary structure. So they take in like the input that you provide there is only the sequence. So once I have my protein sequence, I put it there, and then they help me predict with that if it's a helix or if it's a sheet or if it's nothing.
Okay, okay, Bhaiya, thank you.
So now this will come handy as we move on next. So there are 20 types of amino acids, but there are some differences. So these red boxes that you see here, these are aliphatic amino acids. These are like alanine, glycine, isoleucine, leucine, proline, valine. Then there are some aromatic amino acids, phenylalanine, tryptophan, and tyrosine. There are some acidic amino acid, like which is negatively charged. So we have aspartic acid, glutamic acid. There are some amino acids that are positively charged, that is arginine, histidine in some cases, and glycine. And there are some hydroxylic amino acid, that means they have a OH group, that is serine and threonine. And there is also some sulfur containing amino acids, that's cysteine and methionine. And there is also some amidic asparagine and glutamine. So these classes of amino acid will be very useful as we move on to what are disordered proteins and why they are disordered. And one major classification to remember is the one that are polar and the one that are non-polar. I see some. So let's see. So now what happens if a protein do not fold? So we in the first part we talked about like what the primary structure of a protein, from there the secondary structure. Okay, I see something in the chat. Okay, never mind. So in the first slide I showed that the secondary structure of a protein, it can fold into a folded native three-dimensional structure, which was this. Now what happens if it doesn't fold? So I say there is no problem. A protein does not require a native folded structure to perform its function, which is quite contrary to what the lock and key hypothesis says. So these are, we call them intrinsically disordered proteins. It's something like this. So as I said, what happens if a protein does not fold? A protein can exist in this form. There's no problem with that. And the main feature of a intrinsically disordered protein is they have a flat landscape with no minima. Now, what does that mean? So if we look at the free energy of a protein, and if we look at the conformation, so this is a well folded protein. So a folded protein can have only one native state. So the native state will of course be energetically more favorable. So there can be only one structure that corresponds to the global minima of a folded protein, which means that that's the most stable state and that has the least amount of energy. However, in case of a disordered protein, there is no such well-formed energy landscape like the folded protein. You see, so this is a typical energy landscape for a disordered protein, and it's pretty flat. So what does that tell you? That tells you that a disordered protein does not have any, they do not have any global minima, or that means they can have, or you can say they can have several global minimas, which are all kind of equivalent. And there's so Uvretsky's rule of three. Uvretsky is a scientist who works on disordered proteins, and he once said that 3 encounters with IDPs are needed before a researcher takes them seriously. I see a hand.
Yeah, it's me. What is free energy means here?
You can consider it to be any kind of free energy. For example, you can consider it like Gibbs free energy, for example, here. Okay.
Okay.
Mm-hmm. So what it is, again, like what does a free energy landscape tells you? It's just that a folded protein can have only one global minima, or that means only one native state, which is not
Mm-hmm.
a disordered protein.
Okay.
You don't need to I mean, if you can directly unmute yourself and ask it. I don't know, it see the chat for some reason, and I can't see the hand as well. So yeah, I saw someone raised a hand.
Yeah, I raised the hand. Yeah, my question is that in case of the intrinsically disordered protein, these things are, you are showing that they may be in very low or free energy, but they are not very any specific conformation, not very ordered. So how can it be possible? That means you are saying that they are disordered protein, but they are sometimes that energy is high and sometimes energy is low. Well, I didn't
No,
get
no,
it that in respect of folded protein.
no. We do not compare the free energy. So I have not placed them to compare the app, like the value of the free energy. We don't need to do that. What matters here, like what's your takeaway message from this picture, is a folded protein can have one single conformer as its minima, whereas a disordered protein may have none or may have multiple of them. So for example, let's say this structure, okay, uh, I just, this is a random structure. I don't know, I don't even know, remember what protein is this, but this is a particular conformation of a disordered protein, and it doesn't necessarily mean that this has to be the absolute minima. Like it can exist in some other conformation, but that may again still be the minima. So we do not want to compare the values of the free energy. Like the free energy of a disordered protein, it may be greater, it may be less. That, I mean, that doesn't affect us in any way. What affects us is that the lack of this absolute global minima. So you see, for example, in this picture, we have three shallow regions, which means that there are three possible lowest energy conformations here.
Okay, okay, I get it.
Souvik Dey,
Yes.
actually, I was having a bit doubt. Are the do you mean to say the all the minimas are basically of same value? Means all the minima have the same value, sort of.
For a
That is
yes.
for a disordered protein. Okay, that's why in which of the states it will be, it's not really predetermined, sort of.
So, yes, they do not have like a global minimum per se. They can have several of them. Like it can exist in so many different conformations that you do not know that which one of them is the actual global minima.
Okay, means all the minimas have equal value, sort of thing,
Yes, kind of. They are very
or
few.
nearly same, something similar. Okay, okay.
Should I move on?
Okay, it's fine. Move on.
Okay. So now let's see that why do you think a protein would not fold? So in general, a protein, like the most of the proteins that we read in our school or in the first or second year of our undergraduate, we see that they generally fold. But why do you think a protein would not fold? So again, as I said, it all comes down to the amino acid sequence. Now, remember when I classified the amino acids as polar and non-polar. So what happens is, like these disordered proteins, they have too many polar amino acids. So just look at here. So this is an isolated protein. We call this red one as the hydrophobic region. So the hydrophobic means which are not polar, and this blue one is the hydrophilic region. So what happens is, like the basic governing principle for a protein to fold is the hydrophobic region. They kind of cluster together in middle of the protein, and we have the hydrophilic or the polar amino acids at the periphery. So that means if you put a folded protein in water, you can easily reach this state. So the hydrophobic states will, of course, they do not like water, so they would like to be away from them. And so they will kind of like, they will, so that's one of the major factor that helps a protein to fold. However, what happens for disordered proteins is disordered proteins have too many of these blue polar amino acids, and they have too few of the non-polar hydrophobic amino acids. So that's one of the factors which doesn't allow these proteins to fold. This is an example that I picked up from this paper, that this says that this is the all the amino acids that you have, and this is disordered protein minus PDB. So the PDB is a collection of folded protein, and this prot is a collection of disordered protein. So what these people did is they tried to calculate the differences between the occurrence of each amino acid in the disordered protein database and the folded protein database. So if you have a positive value, that definitely means that that particular amino acid occurs in the disordered proteins more. If you have a negative value, that means that particular amino acid is present in the folded protein. So now, as I said, if you look at what all amino acids are there, you will see all of them are the charged amino acids. So the first is, of course, proline. The proline, as I said before, it's a very, it doesn't form an alpha helix. It's a helix breaker. It rarely forms a beta sheet too. Then you have glutamic acid, you have serine, you have lysine, alanine, glycine. On the other hand, if you look at the protein that forms, that are favored in the PDB, those are mostly the hydrophobic or the non-polar amino acids. So you have this aromatic, you have tryptophan, you have phenylalanine, tyrosine, isoleucine, valine. So that basically tells you that there are some amino acid that favors the formation of disordered proteins, and there are some amino acid that favors the formation of ordered proteins. And definitely you can differentiate between the two by looking at whether the amino acid is polar or non-polar. Then IDPs also have a very high net charge. So what happens is if you have a protein with too many charged amino acids, they kind of repel each other. So that's one of the factors that prevents them to fold. One of the major classification of disordered protein is they should have a high net charge and they should have a very low hydrophobicity, which means very less number of hydrophobic residues. So this is one such schematic. So this is on the x-axis you have the mean hydrophobicity, and on the y-axis you have mean net charge. So if you see, if we try to draw a line between in which cases a protein forms disordered protein and in which case it forms a globular, that means a structured protein, you see most of the disordered protein have a very high mean net charge. So mean net charge is basically the net charge of the protein divided by the total number of amino acids. So most of these disordered proteins have a very high mean net charge and also very low hydrophobicity. Whereas if you see, there is only one such example though of a folded protein, but that has a very low net charge and also the hydrophobicity is pretty high. And then another factor that also favors the formation of disordered protein is something we call low complexity domain. So low complexity domain occurs when you have the same, like you have repetitions of the same sequence multiple times. For example, let's say you have a glycine and a serine, and you have the same glycine serine repetition multiple times in a protein. That also it kind of does not allow a protein to fold. And again, like as I said, intrinsically disordered proteins have too many prolines in between them. So a proline does not allow an alpha helix to form, and if an alpha helix is formed, it breaks the helix and it rarely forms any other secondary structure other than it rarely forms something that we call polyproline 2 helices. It's similar to alpha helix, but it's formed by prolines, and that's quite prevalent in disordered proteins.
Hello.
Yes.
Yeah, you told that this polyproline is quite similar to the alpha helix. So that means it has a definite structure.
Not like, so the alpha helix, we define secondary structure as their position on the Ramachandran map. So polyproline helices also has a definite like region on the Ramachandran map. However, they are not as ordered as alpha helices are. It's a very like, if you look at polyproline helix in the literature, the degree of order will be much lesser than alpha helix.
Degree of order means
I mean, so an alpha helix is a pretty ordered secondary structure, right? You mean you see a very like well repeated region, and it's also usually
okay.
a polyproline helix can be
Okay.
in like 3 residues.
Okay.
You can start from as low as 3 residues.
Okay.
You won't find an alpha helix. I mean, you can, but it will be very rare. You just find that less.
Okay.
Okay, anything else? Then I think.
No.
Okay. So we say that IDPs are born to be wild. Now, why do we say that? So typically for an ordered protein, you know that that's a very well-known phenomena, that if you heat a protein, like if you make it go at a higher temperature, they lose their structure. However, what happens is, in case of certain IDPs, they gain more ordered structure based on temperature, and they are usually more structured at high temperature. Now, why do you think that happens? So if you increase the temperature, there is an enhancement of this hydrophobic interaction. So before, what was happening, I showed you here. So these red boxes are the hydrophobic residues. This blue are the hydrophilic residues. So if you put a disordered protein in water, now say a disordered protein does not have a folded structure. It has it's kind of this kind of a structure. So if you have too many polar or hydrophilic residues, what happens is they interact with water, and as a result, they are not able to fold. But if you heat, like if you increase the temperature, what happens is the water molecules, they kind of move away. So in that case, the polar amino acids are no longer connected with the water, and in that case, that favors kind of folding. And this has been observed in many IDPs, but some pretty well-known ones are alpha-synuclein and casein. This casein is the one that's present in the milk. A similar effect can also be seen with pH, with the net charge being affected in that case. So we are trying, if we try to alter the two main features that helps in the formation of a disordered protein. So I talked about two main features. One was it should have a high net charge, and also it should have a low hydrophobicity. So if you try to increase the hydrophobic interaction, or if you try to play around with the pH such that if you can reduce the high net charge, in that case, IDPs can form, like there might be some tendencies for them to fold. Okay. IDPs are also, they are known to be interaction professionals. So what that means is they can interact with so many different proteins, lipids, and other things. So as I already said before, IDPs, they do not fit into the lock and key model of enzyme activity. So the lock and key model, again, it says that you need to have a predefined structure for some things, for some protein to fit into some other protein. So, however, we don't consider that as a problem. IDPs are always forming static, semi-static, fuzzy, or dynamic complexes having high specificity and low affinity with other proteins or lipids. They may even undergo folding upon binding transitions that we call the induced fit model. This is an example. So this is a protein called ACTR, that's the activation domain from P160 transcriptional cofactor. And this is NCBD, this is a nuclear coactivator binding domain. So if you look at their native structure, this one doesn't have a native structure, both of them. However, what happens is if when they combine, they give rise to a folded structure. That means there might be a protein which is disordered in the native state. However, they when IDPs, they bind with some in, like, for example, let's say an IDP bind with another protein, another structured protein, they can become structured. So these things have been observed multiple times in the literature, that a disordered protein may undergo folding upon binding transitions. So we used to call it the induced fit model. So when we learned about enzyme activities, there were two models that that were talked about. One was the lock and key model, in which this protein certainly cannot fit. There's also another one which we call the induced fit model. That means a protein will first look at what kind of active site is there, and then it will modify itself accordingly so that it can fit in that place. And that was the basis for the disordered protein. I mean, that's what we call them induced fit. So that's very much well represented in this example. Both of these proteins, they didn't have any secondary structure before, but as they like, so this is ACTR in complex, is NCBD in complex. They are, if you look carefully, these two structures are same, which means this binds with this. So before, none of them had a structure, and once they bind, both of them, they have a pretty well regular folded state. So this kind of interaction IDPs are regularly forming with other proteins. Now, one of the most, you might ask that why disordered proteins can bind with several other things. So again, their structure favors it more. A disordered protein does, like as I said before, it does not have a well-defined structure. It can have any structure it wants. That kind of helps them to interact with multiple other partners, which would not have been possible if the structure was the same. Is there any questions, or if not, I can move on to that.
So in that case, which promotes to this or increasing the order of this protein? The last time, that is that increasing hydrophobicity or something, changing of pH. In that case, when it is bind with another protein,
So
which promotes it?
So if you ask that what makes an IDP to go into an ordered state when they bind with something, that's again depends from case by case. Sometimes what might happen is an IDP might let's say when it interacts with another structured protein, what might happen is there might be some specific interactions. For example, there might be some interaction with like amino acid specific, and that can help it to fold. And again, like I said, like it depends from case, it depends very much on a case by case basis. There is, I cannot say that there exists a general framework why this happens, but this has been observed pretty much in the literature. It's
you mentioned that proline based proteins are less subject to go into an amino acid, go into an alpha helix configuration.
Yes, so always you can mostly find prolines at the very end of a helix. So prolines are well known to be helix breakers.
Right. So in case of two IDPs interacting, do they form alpha helixes, or are in general alpha helixes avoided?
They may form alpha helices. Like as I said, there is no like fixed protocol that they have to they have to form a particular kind of structure. It varies very much from a case to case basis. They may form alpha helix, they may form beta shapes.
in those cases, if they form alpha helices, is the proline domain kind of taken
No.
out of the picture?
So an alpha, so the basic, so the fundament, the way in which alpha helix is formed, that does not change here. So you will not find a proline in the middle of an alpha helix because that will break down the helix in any case.
Okay, so it's usually, even if proline is present, it will be towards at the end of the alpha helix.
Alpha helix, yes.
Okay, right.
Okay, then let's go ahead. Now, one of the most interesting studies in present days is the formation of membraneless organelles in cells that are dominated by IDPs. Now, you all know that not all organelles in the cell has membranes, for example, ribosomes, centrioles, etc. So, how do you think they can maintain their shape? So, in organelles so all the cell organelles are present in the cytoplasm. And in case you have a cell organelle which has a membrane, you can think that, okay, the membrane kind of maintains the shape of that organelle. But what if you have certain things like these ribosomes and centrioles? How do you think they can maintain their shape? So these are the manifestations of some phenomena which we call liquid-liquid phase separation that are moderated by the disordered proteins. Now I'm coming to what happens there, but a basic picture is, for example, you consider this is a cell and this is a membraneless organelle. So what happens is this is formed basically by the formation of two phases inside a liquid. So this is a single phase and this kind of forms into a droplet. So a droplet is basically a more condensed liquid, you can say. This is an image of how they look like. So what happens here, this is HNRNPA1, and this is a microscope image of a droplet that has been formed. So if you look carefully, these are nothing but this is a liquid within a liquid. So a liquid can have two different phases, one's dense phase and one is like not so dense phase, and they undergo a phase separation to give rise to these membraneless organelles. I am coming to what happens there, but this is the basic fundamental of why we are interested in this liquid-liquid phase separation. So so liquid-liquid phase separation, what is that? So let us consider a mix of ice and water. So let's say you have a glass in which you have both ice and water. So you know that they can coexist, right? How can they coexist? They coexist because they have the same free energy. So this is the basic definition of a Gibbs free energy. That's delta G equals delta H minus T delta S. So you can achieve, if you look at the equation, you can achieve a low value of the free energy by two ways. Either you decrease the value of the H or you increase the value of the S. So if you have a solid and a liquid state, a solid will have a low enthalpy and a liquid will have a high entropy. So either you achieve the low free energy via enthalpy or you achieve the low free energy via entropy. Either of them works. A protein can also you can also see a similar thing in case of protein. So what happens is in proteins at temperature below a critical value, the solution phase of the proteins can further separate into a dilute phase and a concentrated phase. And the protein molecules are liquid-like in both phases. So just imagine that there was a protein solution before. And now when if you keep reducing the temperature, a time will come when you will go below a critical temperature that the solution phase of the protein will separate into two different densities. They will separate into two states of liquids which have different densities. So what happens is that in the concentrated phase, you have protein molecules, they condense to form droplets and they scatter light and they appear cloudy to the naked eye. So the picture that I showed you before in the previous page, these are the formation of droplets, which means they are the high density region in a disordered protein. So that's what we call the concentrated phase. So in the concentrated phase, the protein molecules, they condense to form droplets. So naturally, if you have a high density region, you will scatter light and it appears cloudy to the naked eye. It's the same principle by which you see the clouds in the sky. So now, why IDPs favor this phenomena, liquid-liquid phase separation? It's because IDPs do not crystallize, and hence for them this is thermodynamically stable. So there is no competition from the solution to solid phase separation, and hence it's easily observable. So there have been many instances that have been shown that the basis of the formation of the membraneless organelles inside the cell is governed by this liquid-liquid phase separation phenomena, and this liquid-liquid phase separation in turn is enhanced by the formation of like IDPs. They generally favor the formation of liquid-liquid phase separation, or as we say, the formation of membraneless organelles. Any questions on that? Or is it, or did I make it too hard, this part?
Yeah, I think no, you can proceed.
Okay. Yeah. So this is my last slide. So what do I do? I basically study the dynamics of IDPs by theoretical method we call the molecular dynamics simulation. Now coming to dynamics, so if you have a well folded protein, a folded protein can have dynamics at two different scales. One is the global motion, that is the movement of a particular domain of a protein, like they move as a whole, and there might be local motion, that's just bond vibrations. However, for IDPs, it's not so simple. So you have different time scales of motion that are not so well separable like a structured protein, and they are pretty much intertwined. Like you cannot separate them so nicely. So what we do is we study the dynamics of IDPs by molecular dynamics simulation, and we try to identify the sequence dependence of IDPs using both theoretical and experimental methods. So we start with a sequence, we do the molecular dynamics simulation. Now the simulations are based on a very simple principle, that's Newton's law that you all know. So in Newton's law, we define force as mass times acceleration. So here also, what we do is we have the individual protein molecules, the individual amino acids, and then we apply force and we see that how much, what's the acceleration of that. And I'll show you a video of what empty simulation looks like for an IDP. You can see, so sometimes when we do IDP simulation in water, sometimes we see the formation of structured element as well. And then we try to identify the sequence dependence of IDP dynamics. So what sequence dependence means? That means if we can identify that, if we can identify that certain IDPs are moving faster than the other, we try to know why, like, or if something's moving slower, we try to identify what's the sequence behind that. We can do that either by using molecular dynamics simulation or by using NMR. So NMR is again a very vast technique. I'm not going to go into that, but we do some kind of NMR studies. Let me see if I can show something. Give me 1 second. Can you see another screen now?
Yeah.
Yes.
So this is a video made by a professor in University of Maryland. He made a really nice video showing a MD simulation, and he mixed it up with music. And you see, this is really beautiful. So let me go back to my PowerPoint. So what we try to do is, like I showed in the video, we have a disordered protein. We try to apply Newton's law on that, and we try to see how that moves and at what speed they move. Like if there is any specific dynamical information that we can extract, and that becomes particularly useful if we go on to study protein-protein interaction, protein-lipid interactions, and so on. So that's all I have, and I'd like to thank you all for your attention. Now, if there is any question that you have, anything at all that you didn't understand or you would like me.
Is this the particular protein, the one you're showing right now?
This one?
Yeah.
It must be. I just took it from somewhere. I don't know what protein it is.
Right.
I have a question here. The flatness that is appearing in this picture and the picture you and the video you just saw. So is it due to the feature of like some simulational feature, not the viewing angles, right?
Which one are you saying? This is just a random image. So, and that simulation that I was showing before, that's different. These are not the same.
Yeah, the flatness I was telling about. Even in the video, there is like the flatness area. There's this
okay,
thin line.
1 second. Let me just go back to there. You mean this one?
The yellow region.
Yeah.
So here it is gone, right?
Yeah. So you mean the flatness as in what? Like, so in the yellow region was actually forming a beta sheet. So if you have a beta sheet, they will be always parallel to each other. So in that case, they will be kind of flat.
So that's just a viewing angle.
Yeah. That was just because of the formation of the beta sheet. So a beta sheet can only be formed if you have this in parallel to each other. Otherwise, so you see in all these times they do not form anything, but only when they have those yellow regions. So the yellow is the beta sheet. They will have like you will have that arrangement, like the parallel thing that you are talking about.
Thanks.
Okay. Does anybody else have anything else, or I should assume that I have made things very clear to you all? No other questions? Then maybe I should just stop the sharing.
One thing is that like from all these molecular dynamic simulations, what is your overall goal? What's the goal that you're looking to achieve, or what's your objective?
So that's what I was saying. We try to use this molecular dynamic simulations. We see those like how these proteins are moving, right? We try to identify the different dynamic scales. Like an IDP can have various types of motions going on. Some motion can be at the level of picosecond, some may be at the level of nanosecond, some may be tens of nanoseconds. So we try to understand and isolate those motions from each other. And then we use NMR to see how like we try to identify that how can we use the simulations with NMR to understand the dynamical scales of these IDPs. Like we try to identify if some amino acid in a disordered protein is moving too fast, if something is moving too slow, something like that.
Are you trying to find out a kind of a functional relationship, or is it a qualitative relationship?
I mean, we are trying to so basically we are trying to develop a global scale, like something that fits all IDPs, and something that has to do with sequence. So we are trying to see if some particular amino acid can have like some amino acid.
শুনতে পেলে সৌভিক?
হ্যাঁ।
So are you kind of looking to find out what features of an amino acid promotes IDPs, or promotes certain characteristics of IDP dynamics?
Yes. So we are trying to identify, first of all, we start with the NMR relaxation. So you must have know like NMR can give transverse and longitudinal relaxations.
Yeah.
So based on that, we try to see if the NMR relaxation is sequence dependent. So if some particular amino acid is giving rise to a high relaxation rate, if something is giving rise to low relaxation rate, and then we try to understand that why is that happening? Like why would something have a high relaxation rate or low relaxation rate? In like even if we talk in general, like if we forget relaxation rates are nothing but they just show how well one residue interacts with another. Quantitatively, in a very broad sense, it comes down to that. So we try to understand if there is some amino acid dependence, or if it's completely like random in nature.
Okay, I see. Okay.
So is there any other question? Then let me just stop the sharing.
Yeah, you just told that this IDP is mainly present on the organelle which has not any membrane. That means only in ribosome. So that is that any effect of like that this mechanism of cell signaling or something like that, or anything? So
I didn't get your question, sir. Could you say that again?
yeah,
no,
my question is that
just a second. So before that, just let me tell you. So IDPs they are present in membraneless organelles, but they can also be present elsewhere. Go ahead.
okay, no, that means is that any role of IDP in case of any receptor or something, that cell signaling or any function of any hormone or something, this thing, or act as any mediator or action in transport or something, then this can affect another biological activities in our body. So, or any cell. So, is that so? That then, what is the thing?
You mean if IDPs can help in cell signaling? I guess so. I'm not so sure, but
Yeah.
I think it should, because IDPs can like if you were talking something about transporter or something, IDPs can bind things very easily because of its structure. It can bind things very easily. It can also, like, if you change the structure, naturally it will just unbind as well pretty easily. But I'm not so sure about the signaling part. I have never looked into that.
That means we can externally manipulate this thing.
Yes, yes. So their structure is very dynamic in nature. Like if you change certain conditions, their structure will change very, very much. Like as I was saying, it forms very fuzzy complexes with lipids and other proteins, and they are very kind of random in nature.
Okay. Okay, I get it. Thank you.
How much weight have you gained?
I think 4 to 5